Hello, and welcome to the ParAqua-COST, the science podcast that shines a light on the newest and most interesting research being conducted in the fields of aquatic and marine ecology. My name is Joe Money, I'm a PhD student and the host of this podcast. Today I'm joined by Martyna Budziak, a PhD student from the Institute of Nature Conservation, which is part of the Polish Academy of Sciences, based in Krakow. Together we'll be discussing her research into microplastics, cyanobacterial blooms, and lesser known uses of algal products. Martina, how are you doing?
I'm great. Thank you very much. How are you?
Yeah, not doing too badly. I've heard somewhere that you have been doing fieldwork this week, and last week.
Oh,
How
yeah.
has it been going?
It was great, honestly. Perfect weather for sampling, but as always, a lot of work, but very rewarding. Thanks.
Yeah, they are tough, these fieldwork samples if you're working with lakes, but much better to do it in August than to do it in, let's say, January.
Oh, yeah. We do that in January too. So, yeah.
Yeah, it can be tough, can't it?
Mm.
So, Martina, could you give us a quick summary of your background, your education, and your research history?
Of course, I would be very pleased to do that. I'm from Poland, as you mentioned. I'm a PhD student based in Krakow. Generally, I grew up in the south, so somewhere around this mountain area, Tatra Mountains. And I think that's partly why I love nature so much, because I was always surrounded by it with everything. But also, I grew up in a very, very small village, so I was itching to see the city to move out. And that's how I ended up in Krakow for my undergraduate studies. And for my undergrad, I did biology. For my master's, I did environmental protection and management, both of them in Jagiellonian University. And during that time, I was researching microplastics and their connection to ciliates. And after that, I started my PhD in the Institute of Nature Conservation. And here I am now.
Nice, nice. I actually didn't know that you grew up in the southern mountains of Poland. I visited there a little bit, and it is really, really beautiful. And
That's
wow, yeah,
true.
undergrad and PhD in Krakow. That's quite cool. And another amazing city, almost as good as Poznan, in my opinion, I would say.
Oh, yeah, almost. Yeah,
Yeah.
definitely that.
Nice. So you mentioned microplastics, and they have become a bit of a hot topic in recent years, not only in science, but also in public discourse, as more and more people seem to realize just how prevalent they've become in the environment. So could you tell the listeners a little bit about your research involving microplastics?
Yes, yes, of course. And I absolutely agree with you. Microplastics nowadays, especially, they're a big concern because they're these tiny, tiny size, which allows them to be eaten by aquatic organisms and then to travel through the food chain, potentially even reaching humans. Well, now I think there's still very little evidence on the effect of microplastics on people, but as I like to say it, to understand the whole story, the end of the story, it is crucial to look at the beginning. And and obviously that's where ciliates come in. They are single-celled organisms. They are very, very small and covered in tiny hair-like structures called cilia. That's why ciliates. And they use them to move around, to catch food. And so basically, they are very, very small and very, very hairy, but in a cute microscopic way, of course. And uh, to like put it into perspective, I think they would be smaller than a grain of sand. But their role is not so small because they act as a key link in the food chain. They transfer energy from bacteria because they feed on bacteria up to higher organisms. And because of that link, they can also transport microplastics to other organisms, as we know that microplastics could be the size of bacteria, for example. So with the research, we wanted to understand how these tiny, tiny ciliates respond to pollution in their world, because microplastics could affect their feeding, their growth. And to do that, we set up a lab experiment with three different species of ciliates. We exposed them to these tiny microplastic beads, but there were different sizes, different concentrations of them. And of course, we wanted to observe over time how many particles could be ingested, whether it affected or not on the growth of the ciliate. So the goal was to understand how something small can affect through the aquatic food web.
Yeah, so they're a really good example of a good study species to test the effects of microplastics on a single cellular organism. And maybe from there we can extrapolate those effects to how they might affect animals or humans, for example.
Yeah, absolutely. I agree. This is a great point because ciliates have many, many advantages as experimental species.
Hmm.
They're very small, so that's an advantage there. They have very short life cycle. They're relatively easy and cheap to maintain. And also, what is very nice, there's a possibility of cloning. So you can perform the experiment on genetically identical clones. These advantages of ciliates made them very useful in research, in all of the lab experiments, even for toxicity, and also for microplastics. Yeah.
Nice. Very cool. And what were the effects of these experimental exposures on these acidiate species? And what implications do your results have for organisms higher up in the food chain, such as humans, for example?
Well, we saw that microplastics affected different ciliate species in different ways. So the response was species specific. Some species growth slowed down in higher concentration, while the others were less affected. At some point, we even saw that these on the short time exposition to microplastics, we could even see a hormesis-like effect, which means that the microplastics were beneficial. And the number of particles they ingested, it was also very much dependent on the concentration, on the size of the bead, and also on the time exposure. And so the feeding efficiency, basically how well they capture food, dropped in conditions with more microplastics. And what is also important here, I think, is that they did ingest microplastics, but they didn't digest it. So after about an hour, the microplastics were excreted from the cell in form of these compacted clumps. And because ciliates temporarily store these particles in the vacuoles, obviously, while feeding, it shows this potential way where microplastic could be passed along the food web. So if these ciliates would eat the microplastic and then be eaten, obviously we can see how the microplastic travels. However, it's difficult to draw a definite conclusion about effects on humans or like higher organisms at this stage, because for me, I think it would be hard to distinguish between microplastics that were moving through the food web from these lower parts up from those that we were exposed to directly. Because as we know now, microplastics are everywhere, not only in the environment, but also in the air and, for example, bottled water, etc. So I believe it just needs more research at this point to understand these long-term consequences and whether these microplastics that were accumulated by microorganisms could meaningfully impact the species further up.
Wow, that's really cool. And actually, that's the first I've actually heard of the sort of aggregation effect that organisms can do with microplastics and the sort of excreting them, I guess, just passing them across the membrane. That's quite interesting results and definitely maybe an area of potential for future research, looking at how maybe we can use organisms to sort of aggregate and remove these microplastics from the environment. That might be a bit of a stretch, but it's just what jumped to my mind immediately after hearing that.
Yeah, absolutely. That's true because we can imagine if this small microplastics, for example, the one we researched were 1 or 2 micrometers in diameter, but then while they traveled through the ciliate, these aggregates were much bigger. So the bigger ones become more available to higher organisms for,
Mhm.
like, for example, invertebrates that are in water.
Yeah, very, very interesting stuff. You then moved from microplastics into research that was more focused on cyanobacteria. Is that right?
Yes, that's right. Actually, I started my journey with researching cyanobacteria during my master's. So on my 5th year at the Agilonian, I got an opportunity to work in a life project, our algae service for life. That's how it was called. And as a poor student, I could not let that opportunity go. And I was hired as a technician in my current institute, in the Institute of Nature Conservation of the Polish Academy of Sciences. There, I met my now supervisor and my great team. So, saying big hellos to Ella, Wojtek, and Edek. And yeah, that's how I started researching cyanobacteria. And then, well, I decided that the team is great and I really like it. And I stayed. And I stayed for my PhD. I'm already on my 2nd year, and I'm still very, very fascinated about cyanobacteria. I think they're amazing.
Nice. Yeah, they are a really amazing system to study. I think they're sort of a bit tangentially related to my subject material, but they still are quite fascinating to me, and I still have loads to learn. So I'm looking forward to hearing more about them. Um, so could you summarize the role that cyanobacteria play in freshwater ecosystems and how blue-green algal blooms, as they've come to be known, have become so well known to the general public?
So, cyanobacteria or blue-green algae, you can use both of the terms. They're often in the spotlight for all the wrong reasons. And they are best known to the public for harmful blooms. But the bloom itself is a natural phenomenon. Under the right conditions, which is high temperature and plenty of phosphorus and nitrogen, cyanobacteria can grow very, very rapidly, and this leads to these noticeable blooms. But of course, cyanobacteria aren't all bad. They are the original photosynthesizers, and they played a key role in producing the oxygen that made our atmosphere habitable. So they are these OG of turning sunlight into energy. And today, algae and, of course, including cyanobacteria, produce a huge share of the world's oxygen. And cyanobacteria alone are responsible for like 20% of it. And they are primary producers. They form basically the base of aquatic food webs. They capture carbon dioxide, convert sunlight into energy, and all of that sustains countless other organisms. So while blooms can be alarming, of course, these microorganisms are crucial players in the ecosystems, and they have been fundamental to sustaining life on Earth for really billions of years. And well, if that's not enough to convince you that they aren't all bad, they actually have natural enemies, chytrids, which are fungal parasites. That's what I will be researching during my PhD. These fungi can infect cyanobacteria and, of course, other algae, helping to keep their growth in check and to show basically it shows how ecosystems often have these built-in balances, even for species that sometimes get bad reputation.
Yeah, I mean, some great points you're making, and it sounds like we do have a lot to thank them for. I mean, ma mainly producing the atmosphere that we rely on to live. Yeah, it's a great point. Even in my own study area, when I'm studying Daphnia and lake ecosystems, cyanobacteria again are the kind of the bad guys because Daphnia struggle to feed on them quite as well as they do with regular green algae. Um, and they don't always have as much nutrient potential and they are often harder to break down. But you're completely right that they have such a key role in ecosystems that people aren't aware of, and they just think of them as these sort of blooming pests, maybe.
Yeah, yeah, absolutely. Well, they've been around for billions of years, so that's how it is.
Yeah,
They had to do something.
very true. Yeah. So these blooms that we've mentioned are famous for making freshwater dangerous to animals and humans, but what is it that makes these blooms so harmful?
Well, so cyanobacteria, as part of their survival strategy, they're producing a range of chemical compounds. They can be called secondary metabolites, meaning that they aren't directly involved in growth or reproduction. They like different metabolic pathways. And to be honest, scientists aren't really sure why cyanobacteria produce all of these compounds. It's probably a mix of reasons. Some say that they help defend against grazers or against fungal parasites. Others suggest that it may be protection against UV or like oxidative stress, or maybe they just act as chemicals for communication with their own populations or with something outside. So basically, compounds are like a Swiss Army knife of survival tools. So under certain conditions, some of them can end up being sharp, so harmful to humans and to animals. Of course, some of these compounds are toxins, and they can be harmful to humans and to animals if they end up in water. They can affect liver, they can affect the nervous system or the skin, of course, depending on the type and on the exposure. But again, it's not all bad news. I will play the devil's advocate here. Research is increasingly showing that some cyanobacterial metabolites may have beneficial properties, like acting against viruses, acting against fungi, and of course, they are showing potential in pharmacology. So it's a reminder that these tiny organisms are full of chemical surprises, some dangerous, some potentially life-saving, let's say.
Wow, that's really cool. I actually didn't know that there were so many potential uses for these, what I've always thought of as cyanotoxins and these secondary metabolites that they produce. I always thought they were sort of, yeah, used to sort of eliminate the competition a bit more. But yeah, it's actually fascinating that actually less is known about it than maybe you would think. And I guess again, going back to a previous point you made, this is all about sort of ecosystem balance that these compounds are naturally produced by these organisms, but it's just when the populations get too large that they can become a problem. And that can happen often as a response as a result of human action. Am I right?
Yes, of course. That's a very, very important thing that you're mentioning, anthropopressure. So the human mediated changes on the ecosystem. And of course, part of that is climate change, which makes the blooms more persistent, longer lasting, all thanks to the higher temperatures and the nutrients. Yeah.
Hmm. Yeah, I think even people who are not so into aquatic ecology have heard of eutrophication, this process by which lakes and rivers and even the ocean can become sort of covered in cyanobacterial growth and this sort of unsustainable explosion of life that can often then result in a draining of the nutrients and a destruction of the ecosystem by producing these anoxic conditions. Um, yeah, I think that that's a great example of how we are the ones who are the bad guys rather than cyanobacteria being the bad guys.
Yeah,
Would you agree with that?
exactly. Yeah, I would definitely. I'm really happy that you're mentioning that everyone heard about eutrophication because it kind of proves the point that really it is about education and about letting the public know, and not only public but also other okay, not only the public but also the
Like,
government
yeah,
and
like decision
politicians.
makers and yeah,
Decision
yeah.
makers,
Yeah.
exactly, that's the word I was looking for, to let everyone know, and I'm very happy about it. And so yeah, basically.
So, despite the bad press that cyanobacteria get in general, do you think that these organisms still retain an important place in natural food webs? And furthermore, can these organisms provide any important ecosystem services that are maybe a little overlooked?
Well, I think that cyanobacteria often get this bad reputation because of the toxin producing blooms. But the reality is a little bit more complex and a little bit more complicated, because not all species are harmful. There are perfectly safe cyanobacteria, many of them, and they play really essential roles in the ecosystems. As photosynthetic organisms, they capture carbon dioxide, they produce oxygen, obviously, with the net positive impact on the environment. But there's so much more. Some species produce phycocyanin, a vibrant blue pigment used in foods and cosmetics, while others can be cultivated as nutritious feed or to be used as fertilizer. And yeah, so even cyanobacteria that can produce toxins have potential if they're handled carefully. A perfect example of turning this challenge into an opportunity is the project that I mentioned previously. So, Algae Service for Life. And this initiative focused on managing cyanobacterial blooms and nutrient loads in variety of water bodies. And we did that by harvesting cyanobacterial scums and macroalgae mats. And but the project, well, it went beyond simply removing the biomass, because it explored how these material could be repurposed into valuable products. And what we did is that we could use this biomass for biogas, demonstrating this circular economy approach, and also just by being sustainable. Yeah, sustainable management. And well, adding to these practical outcomes, the project also emphasized raising awareness. So, as I said before, about water quality, about the blooms, like potential health risks, of course, but also to promote this wider adoption of the solutions that are sustainable. And well, in practice, it's always a little harder, because it slightly improved the water quality, but for the effect to be more long lasting, obviously it would have to be done longer and on the bigger scale. But still, we harvested the biomass, generated useful products, and of course, well, the project showed that even cyanobacteria, often sees as the villains, they can provide ecosystem services and they can contribute positively to the community and to the environment.
That's very cool. So this project involved removing cyanobacterial mats from the lakes at sort of a large scale to try and improve the water quality. Is that what you're referencing?
Yeah, we definitely did that. So in the project, they were these harvesters were designed and patented to literally be let on the surface of the lake. They were collecting the biomass, and then you can use the biomass for these other products. So you can use it wherever, because they can travel, obviously. with the harvester itself. You can do it on the lake, you can do it from the land. There is one that is specialized for the cyanobacteria. There is the second one that is specialized for macroalgae. So yeah, the project brought amazing results, and I think it's great. And I believe and I hope so that in the future we will see more of it, because the project's results were very, very positive and promising. And I am eternally grateful for being a part of it.
Wow, that sounds really cool. And actually, I've got two questions for you because when you're saying these harvesters that go on the lakes, in my mind, it's some sort of aquatic lawnmower, sort of sci-fi robot that is going across the surface of the lake. Is that what it is? And if so,
Yeah,
what?
so if you want to know what it looks like, you
Yeah.
need to imagine like this golf course car. You
Oh
know what
yeah,
I'm
like a
talking
buggy.
about?
Yeah,
Yeah, but it's just floating on the lake. Yeah.
that's
That's how
very
it looks.
cool. That's very
Yeah,
cool.
it has this, of course, specialized nets to capture the cyanobacteria and to let water go and keep the biomass. And for the macroalgae mats, of course, it has like this special mechanism to collect the algae. But in general, yeah,
That's very
like a
cool. That's very
like
cool. And the second thing that came to my mind is actually that I hadn't really thought about the industrial uses of cyanobacteria. We've had people on this podcast talk about the sort of industrial uses of green algae and the sort of products that could be made of that, even human foods and supplements and stuff like this. But actually, I hadn't ever considered the sort of the industrial uses of the secondary metabolites of cyanobacteria. That's quite amazing. You mentioned biogas, you mentioned in agricultural feed, was it? And maybe dyes. That's all very interesting stuff, and hopefully a future application for this very interesting set of species.
Yeah, yeah, definitely. I wouldn't say if it's specifically the secondary metabolite that is used,
Oh, I see. Yeah.
uh, but well, could be. I can definitely imagine a situation where a specific compound is discovered. It has a promising, very good purpose. Then we can cultivate cyanobacteria just to extract these compounds and for them to be used, for example, in pharmacology or wherever. Yeah,
Yeah.
definitely. I think that's where the future is going.
That's very cool. And that happens all the time in science, doesn't it? You discover a new species, you discover it's got this sort of protein or cellular machinery that you haven't seen anywhere else, and then suddenly someone finds a way to make it work in a lab or in an industry, and suddenly it goes all over the world and is super famous. Like Taq polymerase comes to mind, and as we're speaking about that, but there's so many other examples, I think.
Absolutely. Yes, 100%. I think it's a hot topic, definitely like literally hot for the
Yeah, for
cyanobacteria.
polymerase, yeah.
So, yeah, when someone is researching that, it must be really, really rewarding to discover a new compound like this. And I think it's very important. And it's very nice that the technology is going further and we can just all the time ask questions, but with the fancier tools.
Definitely. And I'm definitely gonna have a look at these sort of harvesting golf buggies that go on lakes a little bit more after this episode because they sound very cool. Um, and okay, so I think we come to our final question of the interview. Um, so we've talked about how cyanobacteria aren't always as bad for natural ecosystems as people might think, but if you were in charge of marketing at Cyanobacteria Incorporated, what strategies would you use to change the public perception of these organisms?
Well, this is a really, really nice question and very creative. Partly, I can respond with bringing up my dear friend. So, hi, Brandy. I'm really happy about that because as I was starting my PhD, and the moment she learned I will be researching cyanobacteria, she crocheted a little cyanobacteria for me.
Oh, wow.
It's a very happy, it's a very, very happy Planktopteryx. It's smiling to me every day on my desk in my office. So maybe we could start there with some plushies or something cuddly, something positive
That's
for
nice.
this unpleasant image,
Yeah,
maybe. And but maybe also I think something more me would be memes, probably. Like I
nice.
can imagine like, sad algae noises because they make the oxygen and the plants or
And everyone hates
trees
them. Yeah,
are getting
yeah.
are getting the good press.
Well, I'm very looking forward to seeing you make those, and we'll try and well, I'll tell you what, we'll definitely feature the crocheted cyanobacteria on our feeds because I'm sure with your permission and with your friend's permission, everyone would love to see that. I think I'm definitely going to put in an order if she starts taking that to market.
Absolutely. I think she'll be thrilled.
Wonderful. Okay. Well, with that, I think we've come to the end, and all that's left to say is thank you very much for being a great guest. I really enjoyed the talk, and I definitely learned a lot.
Well, thank you very much. I am very honored to be here because I know that it's not everyone that gets invited. So I am very, very happy to be among the very great scientists that already were a part of the podcast. And well, fingers crossed for the science, for cyanobacteria, and for everything.
The ParAqua podcast was funded by COST, the European Cooperation in Science and Technology, and is a product of the ParAqua Action Group. If you are interested in joining an action group or just want to know more, please go to www.paraqua-cost.eu or follow the link in the episode description. The podcast is produced by myself and Serena Rasconi, and the Science Communication Coordinator is Anna Gavrilovich. Once again, thanks for listening, and see you next time on the ParAqua podcast.